sponsored by the by the Polk County Community Foundation. And so she wanted to speak to Elizabeth or anybody else in that group to say thank you for their help in sponsoring our speaker. Our speaker today, I think some of you know very well. I can't really express to you what she did for us. About 10 years ago, the folks, I guess it was the folks at PAC Conservancy who knew that we were wanting to put an easement on this land and protect it, introduced us to Jill Newberry. And at the time, she was a professor of botany at the University of South Carolina upstate in Spartanburg, and had been there, was forever there. No, she was their botany department. Without her, it wouldn't exist. And she started coming up here every 2 weeks for a year, walking this property, grabbing a plant, putting it in her sack, walking, putting it in her sack, week after week. And the end result was an amazing collection of plants, a cataloging of over 500 different species on this land, and a collection of 200 of those 500. Not only picked up, but dried, mounted, marked, identified, and given to us here.
At her expense. Every bit of it.
And her time and her energy and her knowledge, priceless. So we are very, very pleased that she's willing today to come and talk to us about Walnut Creek Preserve and what she did in the collection. And Jill, when you get done, I have to say to you that you, not me, I couldn't have, but you motivated Bob to build a dryer. And so he wants your approval and any advice you have about what else we need to do. And so that will be our next phase, is for us to take up the charge, you too, if you want to help, and find the other 300 plants and dry and mounted and identified and put in the collection. So with that, I welcome all of you, and Jill.
Thank you. Well, I have to say, I had a student that tagged along, and she was in a class, so I was being paid for instructing her. So I felt I would be double dipping if I also paid for my services here. Besides it, who wouldn't love to come up every 2 weeks and walk through this property? I mean that's a no-brainer right there. Usually, one is curious as to how one gets into the idea of taking the vegetative world and putting it in 2 dimensions and pushing it into a metal cabinet and closing the door. What would possess you to spend your life doing that? And I guess I have to say it all came about because the town I lived in Wisconsin had a leash law. And my responsibility was to walk the dachshund, okay? Well, you can't get very far away from a tree when you're walking a dog, right? And the dog is sniffing at the base of the tree. Now, there are 2 things you can do. You can look at the dog, or you can look at the tree. Well, I looked at the tree. And so it was a sort of it became a terminal addiction. Once you know one, you want to know another, and then why are they different? And it's terminal. You just can't quit. So that started the whole process. I started out my academic career in medical technology. That lasted about 6 months. The anatomy class was full, so I took the botany class and never looked back. So all of those serendipitous events led to this point in time to a captive audience and to a collection of dried, brittle specimens. We are actually at the very end of the era of collection. Began with the era of great discovery, when new vistas were opened up by sailing ships plying the oceans and discovering whole new floras and faunas. And so one of the books I brought along is about the age of discovery, and it talks about how, yeah, that's fine, no problem. It talks about how collections began. People with deep pockets had thought it very fashionable to have these bizarre collections of plants and animals. And so the first zoos were actually came about by people paying naturalists to go out and collecting in the wild kangaroos and sloth bears and bringing them back and housing them in cages. And the more bizarre your collection, the more prowess you had in the community. And so too with the plants. Gardens wanted new and different and the most exotic possible, and so these plants were brought back, both by seed and also by collections. Artists went with the naturalists frequently. The naturalist was also an artist, and so the color of the flower and all of that was preserved not in the specimen, but in some of the most beautiful drawings of the flora were done at that time. Today, herbariums are out of vogue. The university at Charlotte and in Nashville have terminated their herbarium. They are now housed, one was just unceremoniously thrown out and another was put in a nature center. Others have been delegated to hallways, as they have in the University of Wisconsin. And they are used less and less. The idea of the whole plant is largely gone. The research now, you can't get money to fund a herbarium, to even buy a herbarium cabinet. The science foundations no longer support those endeavors. So we have collections, very valuable collections throughout the world that are being maintained, and a few, like the Missouri Botanical Gardens, are still adding to the collection, but no new herbarium are truly being developed. Now is the age of DNA, and one person put it to me very succinctly. He said, well, if you got a plant, you're only showing the genes that are being expressed, that are actually, and they aren't, it's only about, 10% of the genetic makeup of that organism. So you're only looking at 10% of the data for trying to figure out how it's related to another organism. But herbariums are incredible reservoirs. The one here, you're probably in the smallest herbarium in the world. Without a doubt, probably one of the smallest. But the plants that are there reflect the plants that were here on this land when it was first set aside. So that in 50 years, as the area develops, as you can see how the flora is changing, what is persisting, what has come in since then. So you have a baseline for all future observations here. Not only that, with the collection, you have information. This is climbing fern, Lycoglia is the genus, And it is found only at one spot in the preserve just below the little pond. Lycoglia is a noxious weed, not this particular species, but a very close in Florida. The climbing fern is the common name of it. So that it's going to be very interesting as this area remains preserved. How is the Lycoglia, how's the climbing fern going to, its distribution change? You have documentation where it was. Now in 50 years, it'll be very interesting to see how it's spread. Whether it too is going to become a noxious weed or not. So that's just one example of how and this specimen, by the way, if you looked at it 200 years from now, it'll look exactly the same. It'll still be in 2 dimensions, but it will still have the information down here of where it was found, when it was collected, who identified it, and all of the particulars that go along with identifying this particular species. I'll show you how this is done in just a little while. With the collection, you have both the underside and the upper side of the leaf. You have the fertile leaves and the sterile leaves, so you have all this information encapsulated in 2 dimensions. Halesia, which is silver bell, has flowers and fruits, so you have both of. But the interesting thing about the Halesia here is you have some very, very old specimens, and all the books say the flowers are white. Yours are pink. Yours are pink. I've never seen that in any of the books. White flowers. But maybe when they get very old, are they old specimens that become pink? Or is it a variety of it? You know, to be discovered. We have fruits here. Inside the fruit are seeds. So that if that becomes extinct, you can come back here, get the seeds, and they will be viable. So you have a reservoir of the possibility of reintroducing something should it have no longer become extant on the property. And yet, you also have an endangered species. Well, you have actually 2. I haven't got the ginseng yet, but I will. But, yeah.
Were those leaves eaten when you preserved it?
Yeah.
Not after, okay.
Yeah, that's a good, I'll go back to that question because that's another important piece of information that a herbarium specimen. This is butternut. It's closely related to black walnut, and it has some differences. Both black walnut and butternut have a chambered pith, and you can see that here if you wanted to look at it. They both produce the nuts, but this is a short-lived tree, and walnut is a long-lived tree. This one is being negatively impacted by a fungus, so that in the populations in the wild are disappearing. In the secluded valley of Walnut Creek, some of these are still producing fruits. They're still actively growing and providing a seed source, perhaps for reintroducing it once this fungi has progressed. So here we have an endangered species. Here we have a species that's found only in one location, but what's going to happen to it in the near future or the distant future? All this information is available. Leaves are being eaten. Okay, so you know that there's an insect or something is that insect going to become more and more prevalent? I worked in developing 13,000 specimens at the University of South Carolina in Spartanburg at their herbarium. And one day I had a phone call. they said, do you have this particular plant? I said, yeah, I got it. Did you get it in a cotton field? Yeah, it's a weed in a cotton field in Cherokee County. And they said, could you send us the specimens? I said, well, this is from the University of, I think it was Alabama. I've slept since then, so don't hold me to it. And so I sent the specimens, and they discovered that there was a fungi growing on this weed that was growing in a cotton field. They harvested spores from those, from the fungi, and they used it as a biological control of the weed in the cotton fields in Texas. The thing that's happened is that they were using herbicides. And herbicides tend to be fairly broad spectrum. And what happens is that the weeds that grow in the cotton fields were very close relatives to the cotton. So any herbicide that would have affected the weed would also have affected the cotton. So they couldn't use, but this fungi, which was very specific for the weed, could be harvested, could be used, and the spores of that fungi would be a natural deterrent to the having to put more and more herbicides on the cotton fields. So you just never know what's going to be involved in this bit of information, you know. You have it preserved, you have seed source, you have diversity information. There's an incredible amount of information locked up in those little cabinets there in the two-dimensional world of the herbarium. Herbariums have been developed for a number of reasons. Here it's to document what was here at any point in time. The largest herbarium in the world is in France. It's almost 10 million specimens. And it was developed in the age of discovery, and it has collections from around the globe. The first eucalyptus ever collected was housed there. Kew Gardens, similar environment. They have only 7 million. You know, France is a little bit ahead. The Missouri Botanical Gardens, I think they're at 7 million too. Their specialty is tropical flora, and they have stations down in the tropics where they are monitoring changes and documenting what was there, looking at future. We've got a mini Missouri Botanical Garden right here. Okay. Right. Probably the most interesting one is the Russian one. The Russian have 7,150,000 specimens. And they were largely collected in the last 200 years. And one of their leading geneticists, Vavilov, became very, very concerned about the loss of genetic variation in crop species. He analyzed that 90% of the genes in apples have been lost by domestication, because you grow the same thing day after, and in one field is just exactly genetically identical to the next field, so you get the crop growing at the same time, the same color of apple, the same. So all of the genetic variation has been bred out. And he realized that we don't even know where the wild apples came from. So not only have we lost this genetic variation, we don't even know if we can get it back. So he started going around the world and trying to locate areas where the crops were domesticated and collecting the wild plants in the vicinity so that that genetic variation could be preserved. He collected specimens, he collected seeds, and he went around the world, and this is where he made the mistake. He went to Arizona. And he told the people in Arizona, "You are fools. You are absolutely missing the boat here. You've got so many different food sources that the Indians used that you've just not dealt with, you know. We live in a world of 20 food species, half of them grasses. There's so much more food out there, but we're not taking advantage of that." He said, "You don't even know where these plants came from that you're using today. Where are the wild tomatoes? Where are the wild potatoes? Those are your plants, and you don't know diddly squat about them." Okay? I mean, he was pretty tenacious, and he did that in around 1932. Stalin heard about his fraternization with the West, and he cut off all funding. He put Vavilov in a gulag, and his assistants were deprived of jobs. What happened to those seeds? Famine was rampant in Russia, particularly at the early part of World War II. All of these seeds were stored: rice, barley, oats, and his people were starving. Did they eat the seeds and survive? They buried them. They buried them, and some places we don't even know where they buried them, but they preserved them, and many of them today are in the international seed reserves in Sweden. It's a fascinating tale. What did he do wrong? He was telling us we had our heads in the sand, but Stalin only saw that he was fraternizing with the West. If you want to read the story, and I urge, it's a really, really intensely, it's where our food comes from. It's the story of Vavilov and his exploits. This is, I don't even know, but I've got half of it underlined. So that's how important I think that particular book is. So that's the Russians. So herbariums can have a very specific role, or they can have a generalist role. In the Midwest, most of the herbariums in Iowa, for example, are grasses. Well, there's where do you think many of the seeds came back for planting the shortgrass prairie and the tallgrass prairies? From those collections, and from areas where they identified they were still growing and could go back to that area and see if it was still there. Now, grasses are really fascinating, and they are so important to us, not for golf courses. That's not what I'm talking about. What I'm talking about is oats, barley, rice, corn, millet, all are grasses. They are our starch source. Sugar. It's the most important food group we have are the grains, and all of them are grasses. I don't advise you to go out and eat your clippings, but they are definitely worthy of having a reserve, so that you can reestablish a whole ecosystem based on these reserves and knowing exactly where they grew, because you have that data on the sheet. What I want to caution you about is that there are some problems with herbarium. They're costly to maintain. they take up space, and they bias you in certain respects. Many collectors will tend to, particularly students, they'll take the smallest because it'll fit in the press better, right? So the specimens in the herbarium may be a diminutive form of the wild type. The color is largely gone. The smell is gone. And there is a tendency to over-collect the pretties. All right? So, you're not going to have a lot of tourists going in and looking at the grass collection. It's just not going to happen. And you're only preserving the physical evidence. All the DNA, but you are preserving the DNA. So you do have an encapsulation of the DNA within the seed. And so, you do have that information preserved, but it's not a visual imprint. But the specimens are as permanent as you can get them. The paper is rag. It's old cotton rags, 100% cotton fiber. It's pH neutral, so it will not yellow. It will not become brittle. And it will stay in this kind of off-white color for perpetuity. The glues are water-based. Why would you use a water base? Well, the plant's water-based, so it will go into the tissues. And if this should become damaged, you can take it, put it underwater, and float the specimen up and remount it. We use bookbinding tape, the cotton bookbinding tape to strap it. In Russia, they use silk threads. They have little old ladies that sit there and tie knots underneath and they're beautiful specimens. I mean, my God, the time. Well, I didn't have silk thread, so I used that. The label is cotton fiber. Now, here's a problem I encountered. How do you print the label? You can't use inkjet printer. There's no evidence how long that ink will last. So you have to use the whole carbon, the ribbon of the old typewriters, or the Apple image writers. They use the carbon ribbon. The carbon's going to be around. But these are the sort of things that I called Hewitt Packer. How long are your inks going to last? Well, how long do you want them to last? I said, 500 years. And they go, we probably will. That's not good enough. Now, when you're spending 2 hours to 3 hours on a specimen, and you want to have some evidence that it's going to be around a while. Those are some of the problems. So let me show you as we progress through here. It's an 18th century science, maybe 17th. It's largely unchanged. This is a plant press. Well, looks like any other plant press, except I made it myself because I wasn't going to spend $50 a plant press from